Hello.
Hello, how are you?
Good. How are you?
All right.
Am I visible?
Yeah,
Oh,
I
yes.
can see you.
Nice. Yes.
Great. So, how have you been doing?
Good. Yeah. Kind of like, I missed like some sessions like a week ago and 2 weeks ago. Usually I'm trying to kind of like to arrange it, but it's in work time for me. So
Right.
it's, yeah, kind of like when I can. Kind of
Yeah.
like, I thought like, all right, it's good. But yeah. Yeah, but being fine. Had, and I was like away like last weekend, so yeah, so been like in a dancing festival, so that was nice.
Oh, yeah. That's nice. So, yeah,
Yes.
have you done any more thinking about like some of the Basilaria stuff, or?
Yes. Yeah, can give an update. You want to wait for more people or like?
We can do it now. Yeah.
Okay. Yeah. So yeah, I kind of like divided it from what we talked about with Thomas into kind of like some kind of like micro like task like for me to do. It's just like I took a video and then I trimmed it to like track like one diatom that I want. Diatom. Hello.
Hello.
Just updating on what I've been doing lately. Sorry. Regarding the diatoms in the Basilaria.
Okay.
And yeah, so I took a video, trimmed like a segment of it to kind of track. Because it's moving a bit fast and like it's a bit like zoomed in, so it was just a second or a second and a half. I trimmed it, created a new video. I decided like a tracking point with on the diatom. I'm gonna show it in a second. You can have an image.
Okay.
I activated the tracker to track this point. I kind of like showed on the image like the tracking path. I got the pixels and I saved it to file. And there are still more stuff that I wrote for myself to do before I'm going to send it to Thomas. So he sent me a scaling image. So kind of like on the image of the movie. Hello. Just giving an update on what I've been doing so far in the diatoms. So Thomas sent me like a scaling image. So I took this like 2 pixels like between them and kind of like created like a ratio of kind of like how to transform like each pixel to a micrometer measurement or like a meter. So I got the scale. I'm gonna need to kind of like to apply it to transform the pixels to micrometers, and then I wanted to rotate it to one axis because like most of the calculations that Thomas did were in one axis, and I was thinking how to do it, and lately I kind of just thought of just transform it to polar coordinates, like just a radius and an angle from the center, which is like where the diatom starts to move. And then I'm going to duplicate it to just 2 places within the diatom, and I think that's going to be, ready to be sent to Thomas.
Yeah.
Oh, that sounds good.
Yeah.
Yeah.
Sounds simple enough to succeed too. So, yeah.
Yeah. So it's going to show. Yeah, this is the tracking of one of them. Hopefully, this time the sharing screen is going to work.
Yeah. Here we go.
Is it working?
Yeah.
Oh, okay. Yeah, so you can see like the line, like the tracking line, like it starts like from here. That's the beginning of the movie, and then like it goes. That's the end of the movie, and it's kind of like because it's just a second and a half because the diatom goes from here and then to kind of finishes like already like here, it's just in the end of the field of view. And we have the tracking video. Again, it's very short, but maybe I can. Oh, sorry. And then if I'm gonna do it slower, yeah, you can see like it starts and then just goes all the way. Yeah. That's it.
Oh, that's great. So those lines are sort of like tracking from. I don't understand what the lines are necessarily on the videos, like off the sort of the one end of the diatom cell.
Yeah, let me just share it again. Sorry. Can you see my screen?
Yeah, I can see it.
So the line, it's this point on this diatom, but like the path that the dot did and the tracker did across the different frames. So each frame, it kind of like creates like just like what's the next kind of like line that happened so far, and it prints it on the image.
Okay, Alon, whose movie is this?
That's Thomas. That's the movie Thomas.
Thomas. Okay, that means you see, one problem we have is to prove that the camera is itself not jerky. Okay, and the only way I can think of doing that is to use Stokes' law, put the microscope horizontally and drop something in a liquid and watch it move smoothly and see if it's at even spaces.
Wait, but if with the calculation that Thomas did, don't you think that's gonna help, like deciding if the camera itself is jerky or like just the diatom itself?
I don't know. The problem is to calibrate a camera for jerkiness. You need a smooth motion, and we're trying to look at a smooth motion. So you have to have something that a priori you're sure is a smooth motion. And at this scale, the only thing that you can be sure is a smooth motion would be Stokes' law of an inanimate object. Okay, so I think there's a mathematics aside, I think the only way to do it is to go back to physics.
Yeah.
Okay.
Okay. Yeah. Maybe next time we chatting with Thomas, maybe we can point it out.
Okay. I'm assuming you're familiar with Stokes' law.
No.
No. Stokes' law is the low Reynolds dynamics motion of a spherical body typically under gravity in a liquid, and there's a small period of acceleration if you drop something into a liquid, and then it falls at a constant speed.
Okay. So the law itself saying what the rate that it's gonna expand?
The law itself will, unless it's an extremely small particle subject to Brownian motion, it should move smoothly. Okay, and that look up Stokes' law.
Yes.
S-T-O-K-E. Okay.
Stokes' law. Thank you.
Yeah, I mean it's it would only be covered if you've taken a course in fluid dynamics.
Yeah, I think it wasn't my physics degree.
Yeah, it might have been high school. It might be in high school physics because it's simple. It's simple looking.
Okay, yeah, read it then.
I mean, Einstein used Stokes' law for very small particles to relate to demonstrate that molecules existed.
With Brownian motion.
With Brownian motion, right. Okay, but for large particles, you don't have to worry about it. So large can be typically, so let's say a millimeter diameter. You could drop a millimeter bead of density greater than water in water, and it will accelerate and then go at a constant speed. Okay, and then you can calibrate the camera for its jerkiness. Now, the reason I bring this up is the first Apple cat.
you're speaking, but you're mute.
Oh, I'm mute. I'm mute.
No,
Susan is.
Oh, Susan's. Oh, Susan.
I'm shaking my head, actually. I
Oh,
don't
yeah.
really want to interrupt too much, but 1 millimeter is about the limit of the Stokes law. Particles get too small after that.
Oh, okay.
Well, it might be a half millimeter or something. I have it in the instructions on that system where you have different densities of balls and you're dropping them in a sucrose solution. That experiment, it says the limit of the.
We had a
I can look it up.
The reason I'm bringing this up is that when Apple came out with its first camera, I bought one, and I was trying to use it, I think, on axolotls. and I found that if you dropped an axolotl egg in water, it sank with a very jerky motion. And I tracked it down to the camera.
Oh.
Okay, the camera was worthless. It did not take pictures at even intervals. Okay, so you know, I guess Apple didn't design their camera for me, but okay. At any rate, so a camera, you have to calibrate the camera itself. The only way I can think of doing that is something that moves smoothly. Stokes' law gives you the opportunity to do that. But Thomas may be upset trying to turn his microscope 90 degrees.
Mm.
Okay.
Why is it hard?
It's very hard with some microscopes, yes.
Okay.
Microscopes can weigh many kilograms.
Yeah.
Yeah, Susan has one that's easy to turn 90 degrees.
Oh, yeah.
It's very small.
Yeah.
Nice.
I have a little microscope. You can buy them on Amazon. Just trying to
Nice.
see what I have it.
Bradley, any comments on this?
Yeah, I agree about calibrating the microscope and I like the images, how you've kind of used the technique to get at some of the movement. So yeah, I think that's good.
I mean, they're probably okay with modern cameras, but I haven't seen anybody calibrating modern cameras or how jerky they are. But see, the problem is with human vision, do you know what flicker fusion is?
No.
Flicker fusion is if you see repeating images, if it looks smooth to you, that's called flicker fusion. Let's say you see move. Okay, flicker fusion for humans is about 30 frames per second.
Oh yeah.
Right. So you can't detect this by eye.
Yeah, you can't detect because of the sample rate of the eye.
Yeah,
it's kind of
because
30 frames per second.
Yeah, basically
It's more
the
than
sample rate of the eye. Right.
Mm-hmm. And also, his camera is 100 frames per second, so any jerky movement more than that he also can't detect with his camera.
Oh, okay. If it's okay, sure.
So he's the movie that he sent me. It's 100 frames per second. So I don't know if he has.
Okay, then you have to make sure that if you do a Stokes experiment, the distance moved can be measured accurately between frames.
Mm-hmm.
Okay.
Okay. So I think next meeting when we're gonna chat to him and we can bring it up.
Yeah.
So
Okay. It might upset him.
Yeah.
Sorry. Doing things right is sometimes hard.
Yes. Well, yeah.
Yeah.
Okay. And go take a 100 course. I thought you were an engineer.
Yeah, but I think we didn't do a fluid dynamics and my
You didn't
engineering.
do fluid dynamics. Okay, well, a lot of engineering is about fluid dynamics. And we're in the what's called the low Reynolds number regime. Which
It's a course on Coursera or something.
Nice.
Yeah.
Yeah, I think one thing that I was just thinking lately about to get this line to like a one dimension is to transform it to polaric coordinates, which I think that's gonna.
Why don't you do something really simple? Put a straight line across your screen and just rotate it until it matches.
Yeah, I can do it, but then I want something automatic that's gonna work for all like points and movements. It's gonna happen.
Sometimes doing it right is better before you do it automatically.
Yeah, I think it's gonna be like not so hard, like to transform it to polarity. Something like
you can if you want to do it automatically. There are I forgot the names of them, but they're algorithms for line detection. So you can just detect the long lines of the diatoms and measure their angle.
Oh yeah, also as well, and then I think and then I'm guessing it's gonna just rotating like the image or like the coordinates itself.
Yeah.
We did that.
I was sorry. Yeah, I was just bringing it up for like suggestions from you. So that's great. Like
edge detection and edge detection, and then getting the angles of the edges might be sufficient.
Yeah, angle prediction. Yeah,
because that will tell you the angle. Then you can rotate it, and you should be dead on. Now we're making an assumption here. That is that all the diatoms in the field of view are parallel to each other.
Mm-hmm.
So if you use edge detection, you could also check that assumption.
Yeah, because I can see all the lines and see like what angle they perform like to be them, and then have like a distribution of kind of like what the angles are.
Right.
Yeah. Nice. Sorry, Bradley, I stopped you. What,
we did something similar to this a couple of years ago when we were working with some other images. We used sort of a line approach to align all the diatom colonies in a single direction. So like for we're doing deep learning pre-training of a model. So, you'd need them all in the same orientation to get a good training set. And so it's just a matter, but this was just simple rotating around a line. There isn't, I think polar coordinates would be maybe a bit better just because, you have, I guess you're talking about around a circle.
Yes.
Yeah. So that's pretty much, that would help, I think, to make it sort of aligned because you're actually not dealing with a straight line. I mean, the colonies, as you see, they fold, but they have like this, they fold in a certain direction and they form these V's in different shapes like that. So it's not just a straight line that you're trying to align. You have curvature.
And when you did it, do you have something that's kind of like a script that does it in the repo? Maybe I can search.
I think it might be in the repo. Yeah, if you look around, I don't remember how it was actually done, or it may or may not be in there. I'm not sure.
Okay. Cool. Maybe I'll just have a quick look to see if it's there.
Yeah.
But maybe if you're saying that probably this kind of like just this module gonna be a good module, kind of further on. So maybe we're just gonna make it like within a different file, then just write a bit of documentation for it that people can use it in the future.
Yeah.
Okay.
Yeah. All right. Well, it's great. That's great work, Alon. Thank you for sharing that. Did we have anything else we wanted to talk about? Any updates from Dick or Susan?
Nope.
Okay. So let's see. It's gonna go over maybe the state of DevoWorm. So as you know, DevoLearn is our... Let's see if I can share my screen. There we go. I guess I'm sharing my screen. Can you see my screen?
Yes.
Okay, good. So as you know, DevoLearn is our where we do a lot of our machine learning activities, but we also have data science demos, and we have now DevoGraph as part of this. So this has been cloned from where we were doing the Google Summer of Code work, and we've put it into this space. So now we have a space for DevoGraph. It's not in the formal release of DevoLearn yet, but we're planning on putting it in. So we need to do a new release of DevoLearn. I'm going to work on that soon. So this is, I think, at release 0.3.0. So we can make another release. Or I guess we could make a separate release, like a module for DevoGraph. And we'll see what happens. I'll try to figure out what the best course of action is. But this is now cloned here in DevoLearn. I haven't done anything with respect to the digital microsphere stuff yet. That's kind of still, I don't know whether I'm going to add it into DevoLearn or not. I might do that, but we'll have to kind of come up with a strategy for that. So there's also a web, I think this is newish. There's a web application for DevoLearn, so if you want to use that, that's something you can do as well. So you can install it on your machine using Python here. This is a standalone program. Or you can use the web interface here. And so the web interface is a Heroku app. So I know they've had some issues with Heroku app recently, but it's there if you want to use it. So that's where we are with DevoLearn. Just a quick update on that. Then I have some things that we could talk about here. We talked about Tensegrity last week, and I found an interesting paper on what was that?
So the Heroku app, it doesn't work at the moment yet. You said like there were some issues with it yet.
There were some issues earlier with it working or not working. Then they moved to a payment plan. I don't remember, but it should still be okay for casual use. I wouldn't, if you really want to do something intensive with it, I'd download the version that you install on your machine. But we have the version up on Heroku app to for people mainly to like get used to it. So
cool.
Yeah. So there's this paper on revisiting tissue tensegrity, and I found this paper. I thought we talk about tensegrity a lot. Revisiting it might be fun to do. So this is revisiting tissue tensegrity, biomaterial-based approaches to measure forces across length scales. And this is in APL Bioengineering, so it's a bioengineering journal. Let me zoom in on the text. So the abstract reads, cell-generated forces play a foundational role in tissue dynamics and homeostasis, and are critically important in several biological processes, including cell migration, wound healing, morphogenesis, and cancer metastasis. Quantifying such forces in vivo is technically challenging. It requires novel strategies that capture mechanical information across molecular, cellular, and tissue length scales, while allowing these studies to be performed in physiologically realistic biological models. So advanced biomaterials can be designed to non-destructively measure these stresses in vitro. In here, we review mechanical characterizations and force-sensing biomaterial-based technologies to provide insight into the mechanical nature of tissue processes. We specifically and uniquely focus on the use of these techniques to identify characteristics of cell and tissue tensegrity. So what they're trying to do is figure out what tensegrity actually is in cells and tissues. And they define this, they put in quotes, and they define it as the hierarchical and modular interplay between tension and compression that provide biological tissues with a remarkable set of mechanical properties and behaviors. So this is the way they define it. I don't know if Susan or Dick want to, if you have issue with that definition or not, or if you think that's a reasonable definition.
Tensegrity is supposed to be across all length scales in biology.
Okay. Yeah.
That's what I understand. So.
Okay.
Yeah, Bradley, I think I pointed out in a paper on Archaea, where I did an appendix on tensegrity, that tensegrity can be thought of as going down to the atomic scale.
Yeah.
I mean, the interplay between tension and compression, you could think of atoms going around a nucleus.
Yeah.
So it's in that sense, tensegrity is pervasive and useless.
You mean useless, like in what sense?
Useless as it's a broad concept. Basically, anything that where forces are balanced, so that something doesn't move, is called tensegrity.
Yeah.
Okay.
Yeah. So.
And that can apply at any level: the atom, the molecule, the crystal, etc.
Yeah. So, based on these observed patterns, we highlight and discuss the emerging role of tensegrity at multiple length scales in tissue dynamics. And they say from homeostasis to morphogenesis to pathological dysfunction. So I guess they mean like at the level of phenotype is pathological dysfunction. Morphogenesis is where you're putting together like a morphology. Homeostasis is where you're maintaining a certain state. So this is multiple length scales, but also these different modes of function, I guess. So they kind of go through, they talk about the human body and how the human body is a dynamic and self-stabilizing structure formed through intricate connections between hierarchical building blocks. So I guess the hierarchical building blocks are these parts of the, things that make up the body. So, you have cells that become tissues that become different parts of the body, and so forth. And they build from there. And they say intricate connections. I don't exactly know what they mean, but I guess it could be thought of as a network, or it could be thought of as interactions between things, functional or structural. So the mechanical structure of intra and extracellular proteins, for example, cells and tissues, play a key role in achieving structural stability in response to widely varying mechanical challenges. So again, this is where you have a lot of different things that the body needs to do, and those challenges are met by this sort of stability. So it does seem like it's kind of like, in a sense, it's useless because it isn't really predictive. It just does, it's just kind of like, this is a descriptive definition. And beyond that.
Yeah, but it's better than continuum mechanics.
Yeah.
Describing something. No.
Yeah. So, let's see. So then, it goes, you have these type of architectures that span from nanometer to centimeter length scales. And
See, they didn't go far enough there.
Yeah.
They went to proteins. They didn't go to monomers, which are molecules, and they didn't go to atoms. And then you could also do the nucleus
Right.
of an atom. Okay.
Yeah.
I mean, why do you get radioactivity? Because the tensegrity screwed up in the nucleus of an atom.
Yeah.
There we go. I like it. Yeah. Anyway, like I said, there's continuum mechanics, and I'm supposed to be measuring things using tensegrity instead of continuum mechanics, so it's supposed to be better. more accurate description of how things move. We'll see. I don't know.
Maybe. The problem with tensegrity is have you seen anything in the mathematics of tensegrity where whether or not a structure collapses is anything but a static structure? Susan?
I'm trying to build one, and it collapses. It's got
Yes.
eigenvalues all over the place, and it keeps throwing errors. So I'm going to string it up like a plant cell and put it in a circle. So that, I don't know. Can you do that? No. I'm lagging. There we go, like that, a circle.
One of the problems with the cell state splitter is the estimate is that it has as few as 65 microtubules in it. In which case, if they are in dynamic instability, in other words, the tubules keep growing and shrinking, how on earth do you retain stability of the tensegrity structure? That is yet to be addressed.
Yeah.
Cells are a part of tissue.
Yeah, but then, okay, so the tissue retains the tensegrity structure of the cell state splitter? Maybe. You might be right.
Yeah, well, if they hold up each other, they hold up their neighbors. I haven't done the experiment yet.
Yeah, Bradley, maybe add to the list of potential papers stability of cell state splitters as tensegrity structures.
Oh yeah, yeah.
Yeah, I can work on that.
It's a relatively easy simulation to do.
Yeah.
Because the number of components is not tremendous. Only 65 microtubules. And there are many configurations of presumed cell state splitters, but we have no idea about their stability under stochastic behavior of the components.
Yeah.
Okay? I mean, the problem is when you're doing it at a molecular level, everything's moving.
Right.
Yeah, a few years ago, I tried looking up papers on that, and everybody just looked at a static tensegrity structure and tried to work out the mathematics of whether or not it would collapse.
I don't know. You see my things?
Yeah.
If they are in alignment, where are we?
Yeah, I know.
But they need a twist in order to be stable at any point.
Okay, maybe that's good insight.
Yeah, that's for the honeycomb-shaped ones.
Yeah, but the cell state splitters are planar, roughly.
They're honeycomb-shaped cells. Like that's an epithelial cell.
Yeah, maybe you're right. Maybe you're right. The stability may lie at the tissue level.
And if you have one like mine,
through the junctions.
Yeah.
But if you've ever tried to build a tensegrity structure, you'll know you always have problems because it continually collapses on you.
There it is. Tensegrity structure.
Yeah. Try building that from scratch.
Yeah, I know. I've read the instructions. I have instructions for these things. And you put them together in a line, and then you have to hang on to this part. And
Really?
yes. Yeah, they have instructions for them. But yeah, they're not simple to put together.
Maybe show that for Alon. I don't know if he's familiar with it. This is a toy that was made for toddlers.
Okay.
But constructing it takes a lot more than toddlers.
Nice.
Okay.
And is that like they're balancing each other,
Yes.
with the kind of with the function?
This kind of tensegrity structure, none of the hard parts touch each other.
Mm.
Okay, they're suspended by elastic band, elastic string.
Nice.
Okay, there's Susan collapsing it.
Yeah.
But if you try to put it together, it collapses all the time.
Yeah.
Oh, nice.
That's yeah, and then if you assume those things are molecules and everything's changing shape while you're trying to do it, the problem just gets worse.
Yeah.
And I built one in COMSOL, multiphysics, and I built the honeycomb version,
Okay.
and it collapses by itself.
Yeah.
It's not stable at all, ever.
Oh.
Hard twist on it, then you can get it to be stable sometimes.
Yep.
Yeah. So
when,
I'm putting a ring around the outside to pretend it's in a tissue and attaching it to the outside
Oh, okay.
ring.
That
Like
makes sense.
a plant cell or something. And I
Okay.
think that will stabilize it. And then I'm going to put several in the ring and pretend it's a tissue that goes on for quite a length.
yeah. So
Okay.
That's my model.
Yeah, we could come up with a program that would be a general tensegrity simulator, which would include stochastic properties such as changes in the length of the components, perhaps motor molecules, which could move things around and stuff like that, and then ask stability questions.
There are tensegrity programs out there. There are. They're not in COMSOL, but there are some, maybe Python, I could find one, I don't know.
Oh, okay.
But they have one of where they made a cell that was stable and more or less like my model here. And then they took out the rods and strings that were not used, that didn't have any tension on them. And they started off with I don't know, more square object, and then as they removed the rods and strings, it became more rounded, which is interesting.
Okay. I'd like to see that.
Yeah, I need to find that.
Okay, Bradley.
yeah.
See, you might be quite interested in this because the tensegrity structure is a three-dimensional network.
Yeah, yes, it is.
Okay, and so we're talking here about the mechanical stability of a network where the nodes and edges could be continually changing under certain rules.
Mm-hmm.
That's an idea, yeah. And use it for something else.
Yeah, that would be interesting. Susan,
Okay.
with that toy model that you had, if you said you compress it, if you compress it, does it come back to its regular shape once it's
Yes.
in that form? So,
Well, yes, as long as the elastics are still retained. The problem is Susan and I have had some of these things for so many years. The elastics
yeah.
not bad.
Yeah, this one is a little loose because the elastics are old.
yeah,
But you can make them with the tensegrity objects with string.
right. And so when you're putting it together, though, it does have a problem of collapsing in a bad way, that it doesn't come back to.
Yeah. If you want to get your own, you can look up Manhattan toy. Gee, what do they call that one? I forgot the name of that one.
I'll get my other one. My new one. Yeah, sure, now it won't come down. I knew there was a reason why I grabbed the other one. Oh. Okay, now I've lost my S hook. Let's see. Squish.
Squishy or something like that. These are for toddlers. It's a toy for toddlers.
Oh, okay.
I don't know if you can see that. Can you see that?
Yeah.
Well, don't have
A
any.
bit blurry, but yes.
Yeah,
Yeah.
squish. Manhattan toy squish. This
Squish.
might be one. There it is.
Yeah.
I give them out as baby gifts to anybody who wants a baby present from me. Usually gets this.
Oh.
Yeah.
Gee, you're very kind to parents. We always gave them the toy that makes lots of noise.
Oh, yeah, they often hate those.
Yes.
I had toys like that around my children, and if they made too much noise, I told them that they had to do that outside where I couldn't hear it, or it wouldn't make any noise anymore. Complete threat.
I put in the chat the name of the other Tensegrity toy made by Manhattan Toys. It's the Wurfel.
Oh, okay, yeah. Yeah.
Okay. And the Wurfel we used as a toy model for the nucleus.
Oh, right.
Because it has many stable configurations, which might represent states of differentiation of cells.
Right. So,
Yeah. It has the same problem.
yeah.
The elastic eventually goes bad.
Yeah. So if we
It
go
wasn't
back,
bad. It lasted.
oh,
They last decades, but that's all.
yeah. Yeah.
Parents really like this one because they can squash them into the carry bag for the kids.
Oh, okay.
Yeah.
Yeah. So if we go
Yeah,
back,
you can't do that.
Go ahead. Oh. If we go back to the paper, they talk about biomaterials and how these can be used in different ways, and that if you have cell forces, self-generated forces playing a critical role in a lot of functional things like tissue morphogenesis, muscle contraction, wound healing, and then dysregulation of these forces often correlate with disease onset and progression. So they're looking at this kind of usefully in terms of this sort of dysregulation of forces. And so let's see if they have any images here. So this is an image here of how in a biological system you have tension and compression. So this is a cell where you have tension acting on a bunch of cells, pushing them together as sort of like a focal point. And then you have, that's actually compression. And then you have tension operating on sort of the rotational orientation of the cells, operating side to side. So if you see these cells as they migrate around in this mass of cells, they experience maybe both tension and compression, depending on their location in the cell body here. And then if you look at the cell, what was that?
That's why I think organoids produce different tissues.
Oh, yeah, maybe. Yeah.
Tensions and compressions involved in a cellular organoid that produces a ball of cells. There's different tensions and compressions as you go inward.
Yeah. Then if you go to a single cell, you see that there are these filaments inside the cell. You have microtubules, and you have these same forces acting on the cell. So it's not just this population of cells. And again, if you can apply or cell-generated forces can conspire to force certain differentiation programs. So there's been a lot of experiments with using cell forces, or putting cells on different substrates, and they differentiate in different ways. So there've been experiments on that. So that can play a role. But you also have things at the single cell level. Yeah.
Yeah. This brings up an interesting thought. Do you know the difference between stress and strain?
Yeah, but I can't give you a definition right now, but I'm familiar with the.
In strain, you watch something over time, and you measure the ratio of the distance between two components.
Okay.
Okay. Now that you can do with much of our embryo data. You can take two cells and consider the distance between them And if that changes, you could call that a change in strain.
Yeah.
Now, the interesting question, strain is easy to measure. In other words, if you can get the coordinates of the centroids of the components,
Right.
for instance, cells in an embryo. The interesting question is whether or not there is a relationship between stress and strain. Stress is the force between the two.
Yes, there's stress strain curves.
Yeah.
And it depends on the material as to
Stress strain curves are typically, they're typically monotonic, and they are also typically linear over significant ranges.
yeah, they're linear within a certain range.
Yeah, so what I'm suggesting is that this you could put maybe on the list of future papers. If there's any way of figuring out to what extent we can rely on strain as a measure of stress.
Yeah.
You have to go in there with some elastic materials that you can use as sort of a guide, sensor.
Yeah, right. There are some very old experiments back, I think, from the 60s by a guy named Rappaport. He measured tension inside single cells, typically oocytes, or maybe fertilized cells. And the way he did it is he put a very fine glass rod into the cell and measured the deflection of the cell, of this rod. Okay?
Yeah.
And so it's not out of the question. That was done, 40, 60 years ago.
In optical coherence elastography, they use gels that they know the exact.
Interesting.
And they use a layering technique where they layer them together, so you can match the movement of the gel to the movement of the tissue.
Oh, okay, neat. Okay, so I'm suggesting then that it may be time to move on and get the stress-strain relationships in embryos and see if we can make anything sensible out of them.
Right.
You can certainly get the strain and the strain maps. You can take a nematode and take any pair of cells, draw a straight line between their centroids, and see how the length of that chain, of that straight line, changes over time.
Yeah.
That would give you a strain map, and then the question is, okay, if we assume it has a relationship to a stress map, if it's proportional to it, can we make any sense of the shape of the embryo over time?
Right. Yeah. That might be quite interesting. And I think we have the data for it. At least we can extrapolate from the data.
I think we do. Yeah. We have the data for it. We just have to apply our brains to it.
Yeah.
Well, you need the stress strain curve for them. I was hoping to do some of that, but I need to finish everything else first, apparently.
Well, how should I put it? If you assume that stress was proportional to strain, and then predicted the shape of the embryo and it came up wrong, you might be able to calibrate the stress-strain relationship by assuming that deviations are caused by nonlinearities in the stress-strain relationship.
Yeah, well, it'll be linear for part of its.
Yeah, it'll be linear, but I'm saying deviations might.
What is the angle? Is it here or here? What is the?
Yeah, well, the whole embryo.
Yeah, so just
the nematode, after all, ends up quite non-spherical, even though it starts out as a heliosphere.
Yeah.
So this might be a good measure of shape of the embryo.
Yeah. So you can measure things at that sort of level of maybe like an embryo or the single cells, or then even down to intercellular junctions in the cytoplasm. Yeah.
Susan, is anyone doing 3D structures of organoids at the cellular level?
They're looking at it, and I have a whole bunch of organoid papers and have ignored them because I'm.
Oh, okay. Because an interesting question would be to apply this to an organoid. See
Yeah.
if your speculation that differentiation is occurring correlates with changes in the stress-strain relationship.
Yeah.
Yeah, I just thought that organoids might be a subject for my experiments, and it would be a great thing to look at. Besides, say, axolotl legs, because there's maybe some people who have organoids at the university that they're discarding, and I could mess around with.
Yeah, there's also, you go back to the old experiments by Steinberg and Moscona, where they simply took two embryonic tissues, made what we would now call an organoid, and watched the change of shape, such as into an onion shape, where one tissue coated the other.
Oh, yeah. Okay.
Okay. Now, I've been following some webinars in a group on soft science.
The SLAM meeting?
Slam, yeah.
No?
Yeah, there have been a few lectures on balls of tissues, but not one of them has gone beyond a ball of one type of cell, which just changes shape, and that's it. Sometimes they'll stick two balls of cells together and let them fuse, but they're all always the same cell type, and they do not watch differentiation. Okay? So this is the physics approach.
Yeah. If you get a large enough ball, you'll have a different pressure inside than you do outside, and you won't have the same type of tissue.
They don't get that far. This is the old physics approach. Given a spherical cup,
Yeah. So this is some more examples here of, actually this is where they're using molecular techniques for measuring cell-generated forces. And in this case, they're using things like FRET, which is a molecular technique to look at forces. And they're wanting to look at like specific things, cell processes, for example, and they're measuring them this way. So there are different ways you can measure the forces. You can look at this whole cells, or you can look at parts of cells. So it's very, then you have these kind of different types of probes. So they have protein probes, which are the FRET protein fusions, and then DNA probes, which are DNA oligonucleotides, that you can use in a similar way. And it just measures local forces in some parts of the cell.
Is there any atomic force microscopy in this paper?
I don't know. Let's see. I don't think they do. I think they're just using these kind of dyes and constructs to measure the forces, but they're not using atomic force microscopy, at least that I know of here.
Okay.
I'm not seeing any.
This looks harder.
Yeah. Yeah, I've seen a lot of people do stuff with FRET, like with whole cells, like if you're just trying to get a handle on like how cells are behaving and you don't have like, high-end equipment, or you want to look at it in bulk, that's actually a pretty good technique for it. But anyways, so this is where you also look at things at the cellular length scale. These are a couple of different indicators here. So you can adhere cells a silicone rubber film and then wrinkle the substrate. So like as I said, you can put cells on a substrate and you can play with the substrate, you can play with the shape or the orientation of it, and you can get the cell to do things. You can actually you can measure the forces, but you can also introduce forces to the cell. So this is where the substrate is wrinkled. In this case, this is where the substrate has like sort of a stippling or something where the cell is moving across it. And then,
Bradley,
yeah.
They seem to have missed patterned substrates.
Yeah.
There's a whole literature on I think Ingber may have started it, where you make a surface which has, for instance, this checkerboard of hydrophobic and hydrophilic regions.
Okay.
And I remember Ingber got the interesting result that the cell, if you made a square, if you made squares, the cells would become square.
Oh, wow. Yeah.
However, this is the old tradition that you don't cite things that are older than 3 years.
I think they were just trying to illustrate the different variety of things you can do, but yeah, that sounds really interesting. Yeah. So then they have a nice table here on different biomaterials for measuring traction forces. So there are things like 2D films, 2D substrates, 3D substrates, 2D micro. I mean, so they have like these different techniques like TFM and micropost. Then they kind of get into some of these 3D matrices, which is, moving closer to the tensegrity structures that we were looking at with the toy models, where you have things in 3 dimensions. Because measuring things in 2 dimensions, it's fine for measuring forces if you want to be really reductionist about it. But in 3D, of course, is where a lot of these structures live. So you can do a lot of things with traction forces in 3 dimensions. You can use collagen, fibrin, or matrigel matrices to sort of do the same things that you do in 2D. I'm looking for more images. Here's something. So this is a figure, techniques for measuring tissue scale forces. So now that we're moving up to the tissue scale, where we have a bunch of cells in a population, and again we have our different types of measurement techniques. So the arrows indicate deformations. You have these cell-laden matrices in A, where you have cell, that they contract due to cell-generated forces. So this is where you have cells in a group and they're contracting and they're affecting each other's shape. And then this shows you a time lapse of this where they contract into a small ball. C is where you have patterned microtissues embedded within a mechanically characterized matrix of fiduciary markers and forces. So you're quantifying the forces using different types of markers and different forces introduced into the to that cell population. So you're pushing them together a little bit differently than in A, but you're ending up with a smaller group of cells that they're contracted. D is actually where you're getting looks like some thin film cantilevers that curl up under contractile forces. So this is almost like the leaf of a cell that's coming out from a bud. So this is like unrolling or rolling up along this axis. So they're contracting or expanding along this axis, and it looks like they're curling like this. They're all on this purple substrate, so that's why you get that kind of shape. And then F is embedded microcantilevers include pre-stress with the self-assembled tissue constructs, and the deflection of cantilevers are correlated with contractility. So in this case, you have these 2 cantilevers, and they're pushing against the cell population. They're introducing contractile forces. So you can see that this is papers about introducing ways to sort of induce tensegrity, but it's really about sort of pattern formation as well, because the argument is that these forces, these types of introducing these forces onto the cells, especially populations of cells, you get this pattern formation. And so these are examples down here, H through N, where you have different forces acting on the cell population. You're starting to get different patterns emerge from those interactions. And so then, let's see. I don't know if there are any other figures in here. I think there's one more. This one here. This is where you have the tensegrity spectrum. So these are stable, dynamic, and unstable structures. So you have this scale at the right. It says tension versus compression. So the purple is where things are in tension, and the red is where they're in compression. And you have these structures. So you start with a sphere, okay? And then you're going to deform that sphere in different ways to, by introducing forces, and you're going to change the shape of it, but you're also introducing tension and compression at different points in that structure. So you can see that in this sphere, it's all compression, or it's, kind of neutral, and then you start to get tension along the edges, but compression in the middle, and then that makes like kind of goes from a sphere to sort of an oblong shape. So it almost looks like an egg where you have an oblong shape. It's kind of like C. elegans we were talking about, where you can deform this spherical shape in different ways. In C. elegans, you start with a single cell, you move to a 2-cell, and then you have founder cells, and then you have more cells that form tissues. In this case, you have this shape, and you're introducing tension and compression in different ways, and you're changing the shape. Yeah.
Give me a minute. I'm going to go to the basement. We just yesterday got a gourd, which would be a real challenge to this.
Okay. Yeah. All right.
Oh, well.
Yeah.
I've been looking at some of this for my paper on Tensegrity.
Yeah.
Where you have compression and tension at the same time in the middle of a sphere. Like you can take it and compress it. And that if you're compressing it this way, I don't know if you can see this.
Yeah.
Compressing it, compressing it. Then you have tension going the other way.
Mm-hmm.
I don't know if you can see that. My camera's really slow. I should almost just shut it off.
Oh, it's fine.
Yeah, we saw it. Yeah, if you kind of like up and down the compression, then like the tension goes like sideways to kind of like.
Yes. So you always get both. They're kind of part of each other.
So this, and then this last one is where you get pathogenesis, which means that, so this first one is homeostasis, then this next one is morphogenesis, where it's stretching out, and pathogenesis is just basically where it's dysfunctional. It's maybe like a cancer cell almost. So it looks kind of deformed. Oh, it looks like Dick's back.
Okay, I'm back here. Let me share the screen.
Let me unshare mine. Okay. All right. Go ahead.
Okay. Trying, it's not responding.
Oh, and recent research indicates by animals that are now extinct. So
looks like Dick's freezing up here. I don't know if he's if he's I don't know.
Maybe disconnected.
Yeah.
Oh shit. I see entire screen. Wrong way. No. No, I'm not getting any luck here. What?
Huh.
Share's not working. Oh wait, maybe it's.
There we go. It is.
No,
it's okay.
now it's working.
Yeah.
Is it working now?
Yes.
Can you see me?
Yeah. But it's it's your browser. So.
Oh, I don't see anything.
Oh.
Let's see.
You're sharing, you see SC, no?
Can you see this board?
Yeah, I can see it. It's in the corner here.
You can see it. I can't see it. That's strange. Okay, this is one.
these are all the same seed too. These are genetic variants.
These are called
autumn wings.
Autumn wings.
All right.
It's a gourd which was apparently eaten by animals that are no longer exist, but is still thing is cultivated by people.
Okay.
Here, I've got a couple more here.
All right.
These are all from the
Here's
same.
one that looks a little normal.
These are all genetically the same type.
Okay.
They're all produced.
Okay.
Yeah.
Like Indian corn. They produce multiple variants.
Here, look at this one.
Wow.
Okay. And one more. There's one more.
Oh, wow. Okay. Here we go.
Okay. They're not, apparently not edible by people, but...
Yeah.
Well, they're edible, but they're very bitter.
Oh, very bitter. Okay.
I want to try.
Anyway, I thought that's a real challenge to the Tensegrity structures.
Yeah, there's so much shape in that, like at multiple scales, yeah.
And the whole surface seems to be covered with tumors.
Is that what they are, or is that just like
I don't know.
what the pattern
Have any
is?
idea if those are plant tumors on the surface?
The lumps appear as the fruit ripens. The young ones don't have them. I don't know what they are.
Okay, that's all we know.
Yeah. Well,
Okay.
yeah.
There was a book I used to have, I don't have it anymore, called The Garden of Gourds. That's in the chat.
Okay.
Which shows some of the crazy varieties of gourds. Gourds were studied for their shape and relationship between shape and cell shape back in the 50s.
All right.
Okay. Anyway, they're fascinating. No one's worked on them. Cell shape of gourds versus time would be very interesting and possibly approachable, because you can do sections of them and get at least cell orientations.
Yeah.
Nice.
That'd be interesting.
Yeah, I was particularly interested. There was one called the snake gourd, which is long and narrow.
Oh, yeah.
How does it become that? Of course, the nematode does that. So
Yeah. Okay, now we're going to talk about some papers on organogenesis and organoids. So we talked a little bit in the Tensegrity discussion about using organoids as a way to look at shape changes in Tensegrity, but this is more about organoids in general. And so there's some interesting papers that have come out in the past week or so on organoids. So let's take a look. The first one, and I give credit to Morgan Howe for finding these papers in the literature. He's very interested in organoids, and hopefully this fosters some discussion on that topic. So the first article is a Nature paper on embryoid model completes gastrulation and neurulation in organogenesis. So gastrulation and neurulation is a milestone in the development of embryos, and it's where you get these changes from gastrulation to neurulation. So gastrulation involves changes in shape, to neurulation, which starts to involve the emergence of the brain and the nervous system. And then organogenesis simply means that you're starting to form organs. So this is nice in an embryo model because it can recapitulate what we see in actual embryos. So these are organoids that serve as embryo models. And one of the reasons why we use organoids is to sort of have a controlled version of that embryogenetic process. So this is the abstract. Embryonic stem cells can undergo many aspects of mammalian embryogenesis in vitro. So you can have embryonic stem cells in a dish or in some sort of three-dimensional matrigel, and you can model a lot of aspects of embryogenesis. But having these cells sort of in a culture, in a disembodied culture, doesn't really cut it for a lot of things. So you can't observe some of these processes that require a geometry. And that's why we want to use organoids to do this. But of course, the developmental potential of embryonic stem cells in culture is substantially extended by interactions with extraembryonic stem cells, including trophoblast stem cells, extraembryonic endoderm stem cells, which are XENs, and inducible XEN cells, or IXENs. So there are three different classes of cell here: the trophoblast stem cell, which is TS cells, the extraembryonic endoderm stem cell, which is XEN, which is for extra, and then inducible XEN cells, IXEN. So the little I is the inducible aspect. Here we assembled stem cell derived embryos in vitro from mouse ES cells TS cells and IXEN cells, and showed that they recapitulate the development of whole natural mouse embryo in utero up to day 8.5 post-fertilization. So that's pretty late in mouse embryogenesis with respect to, having single cells start to form shapes and maybe form organs. So another reason we want to use organoids over just disembodied stem cells is we have all these different cell types that are interacting and sort of sorting themselves into some sort of coherent set of systems. Our embryo model displays head folds with defined forebrain and midbrain regions and develops a beating heart-like structure, a trunk comprising a neural tube and somites, a tail bud containing neuromesodermal progenitors, a gut tube, and primordial germ cells. So you can see that this embryo model that they're building involves a number of different sort of very shapely structures. So you have, of course, head folds, some sort of defined forebrain and midbrain regions. So you have this neurogenesis going on. You have not only the genesis of neurons, but the genesis of the neuronal phenotype. You have heart-like structure, so you have heart cells that are, they're beating, I guess, so they have this pacemaker. And then you have a trunk comprised of a neural tube and somites, so you have the neural tube form with somites, a tail bud with neuromesodermal progenitors. So you have all these different parts of the nervous system that are going to be put together later. Or in this case, just those parts of the nervous system that are being recapitulated. So that's important because we can see the origins of the nervous system and maybe how to engineer that. This complete embryo model develops within an extra embryonic yolk sac. Remember, we're talking about mice, so there's a yolk sac with in a uterus that initiates blood island development. So we have we're actually getting some sort of blood island development here as well. So we're getting these systems put together. So it's not just cells, collectively behaving. They're actually forming these tissues and forming these systems, most importantly. Notably, we demonstrate that the neurulating embryo model assembled from PAX6 knockout ES cells aggregated with wild-type TS cells. So these are the embryonic stem cells, and then the trophoblast stem cells TS. So these are aggregated together. Actually, in the ES cells are PAX6 knockouts. PAX6 are involved in eye development, and then wild-type TS cells don't have a knockout. They're just regular TS cells. And IXEN cells, so these are induced XEN cells, recapitulates the ventral domain expansion of the neural tube that occurs in natural ubiquitous PAX6 knockout embryos. So in PAX6, if you do some experiments, you knock out PAX6 in a normally developing mouse, or in a mouse that's not like this model, but the actual mouse, you can knock out PAX6 and you get a phenotype that's actually, a little bit different than what you'd find in normal development, but you get a recapitulation of this phenotype for the PAX6 knockout embryos. So this is important that we've been able to build this embryo model, show this sort of mutant phenotype, and it's been recapitulated in development, not just in terms of the cells expressing this or that, but in forming these systems. Thus, these complete embryoids, and they call them embryoids as embryo models, are a powerful in vitro model for dissecting the roles of diverse cell lineages and genes in development. So again, we have these cell lineages that are creating these structures, but we also have genes that can be manipulated to modify these structures. Our results demonstrate the self-organization ability of ES cells and two types of extra embryonic stem cells to reconstitute mammalian development through and beyond gastrulation to neurulation and early organogenesis. So this is a nice paper that goes over a lot of this. This is a figure here where they show what they're doing here. They have their different cell types. They're putting them into a common culture. Then they're starting to grow these embryo models. So this is where the shape is starting to take place. So you have this fertilization event, and then you have these different cell types. And then this is where you get the inner formation of the inner cell mass in the mouse embryo. And then you get the different cell types as layers up in this part of the embryo and then around the edge. So you can see the different cell types here labeled by color. And then those cell types then go to when you start to get an embryo, so you start to get changes in shape, over time, you start to get the cell types, but they migrate to different poles in the embryo. So you get the ES epiblast region, you get the TSC trophectoderm region, and then you get all of this like this. So this is the trophectoderm. It's going to transform into this, it's going to unfold into this structure. And then you start to get other types of tissues that form later. So the heart and the gut tube and things like that. And then you start to get a little bit more formation here with a primitive streak. And then you start to get even more formation here. So you have this, you have the trophectoderm up here, the chorion, and some of these, and the yolk sac eventually coming up here. So this would be the trophectoderm and chorion, and then this is the yolk sac. So this is the transformation that occurs in this. And then this is an analysis here of, I believe this is some sort of multidimensional, this is a UMAP analysis of the different cell types. So you can see that I think they're expressing different genes here, or there are different days. So they show the difference between natural embryos and these ETIX embryos. And so you can see that they're somewhat similar but different. So they have this, it's hard to interpret these UMAP patterns, but they basically have the same clusters with the same constituents, more or less, but they're a little bit different in terms of where they're expressed. And again, this is not like specific to any area or region of the embryo. This is just a UMAP analysis and visualization. So you have all these different tissue types and cell types that are put into this different colors, and you can see them kind of clustering in a certain way. So it's similar in a lot of ways to the natural embryos, but there are differences as well. And so this is an example here too of some of, they kind of break this down a little bit more here in F. This is the annotated and combined UMAP natural embryos cultured ex utero and collected at indicated time points, and ETIX embryoids individually labeled and analyzed by tiny RNA-seq. So they're using RNA-seq markers to define these clusters and these types. So you can see that here. So yeah, there are differences and similarities. Then we get into this part of the paper where we talk about they talk about how it develops an anterior brain and pattern neural tube, and I'm referring to the embryo models. So here you have some of the images here where they look at the expression of different genes. So this is E8 embryo here, where you have SOX1 and brachyury being expressed in the embryo. This is, I think, these are different views of the embryo. So this part is expressing SOX1. This part is just expressing brachyury. This is the EITX7. So this is like at day 7 of the embryo model, where you see a little bit of a difference, but it basically recapitulates this pattern of expression of SOX1 and then brachyury. So it's very similar in terms of its expression pattern, but a little bit different in the morphology, as you can tell. And then again, you see this for D. You see OTX2 and FOXG1. You see differences in the morphology, but essentially in the same locations for the expression of those genes. So this figure kind of talks about these different genes and then the different structures that where these things are expressed. And this is in the natural embryo versus the embryo model. So you can see that there's a z-score here where it's a deviation from a mean, either positive or negative, and this just shows like the up or down regulation of these genes in these different tissues or these different structures in the natural embryo and in the embryo model. And you can see that they're very similar in a lot of ways, but also, there are differences in terms of level of expression mainly. It's not really that much different in terms of the direction of expression or the actual expression of these things. So, for example, you see that there's the fraction of, this is the size of these circles, the fraction of cells in a group, the percentage of the cells in a group. So sometimes the number of cells changes, sometimes the level of expression changes, but it's always basically in a similar direction. So that's good. Then finally, we have this PAC6 knockout work. So this is in the embryo models where these PAC6 knockouts, they recapitulate what we see in the mouse embryonic phenotypes. So we have in a mouse, we can knock out PAC6, and we have a certain phenotype that results. It's different from the wild type, and so then we can ask, well, can we do this in an embryo model? And indeed we can, and this is showing the evidence of this. Then they do some annotation here. So this annotation experiment is actually, so this is a gene ontology analysis. So this is where genes are, they have an ontology system where they label the genes with a function. And sometimes the function is trivial, but sometimes it's somewhat specific. So you can look at the biological processes here, these Go terms, and you can get a sense of what genes are enriched for what functions. So, these categories are not they're imperfect, but they give you some sense of what's going on. So there's some genes for movement of cell or subcellular compartment, locomotion, anatomical structure morphogenesis, chemotaxis, taxis, and response to chemical. All those are highly enriched. And so that's interesting because that suggests that there's a lot of motility going on at this stage of development in the embryo models. So this is where you have a lot of these different genes related to movement towards stimulus, chemical stimulus, orientation of the cells, and so forth. So there are a lot of things, and then there are, of course, these genes that are involved in axonogenesis, neurogenesis, nervous system development, axon development. So there are all these gene types that are involved in neural development that are very upregulated as well. And so this is exactly what we would expect out of something like this. We also have, as a final figure, the initiation of gut development. And so this is, again, like the eye and the defined mutant of the eye, we have this gut development looking between the embryoid model or the embryo model and the natural biological embryo. And so you can see that they show the co-expression of genes and morphology. So you can register the gene expression with the morphology, and you see some differences in the morphology. The gene expression is generally in the same place, with some differences because the morphology is a little bit different. And then this part here where they show the same graph where they show the fraction of cells, they show the different, actually in this case it's the different tissues that are forming and the different genes that are being upregulated or downregulated. So this is the z-score again. -2 would be downregulated, +2 would be upregulated. And you can see that there's a lot of upregulation and not a whole lot of downregulation in these structures. And then this shows you the difference between the natural embryo and the embryo model in terms of these different cell types. And then there's a UMAP analysis where you have these structures here, these clusters, and they show this is an overlapping cluster analysis here where a UMAP analysis of this cluster where you show where these cells are sort of differentiating. You have a first heart field, a second heart field, and cardiac mesoderm, and they're all separated out. So you can see that that means that there's a beginning of this structure being formed. So this is the mouse heart, and these are the structures we expect at E8.5, which is developmental day 8.5, and this is what we should be trying, or what we should be achieving in the embryo model, and they seem to be doing that. So there's a lot more information in that paper if you want to check it out. It's a very good paper. This is another paper. This is a little bit different. This is Inferring and Perturbing Sulfate Regulums in Human Brain Organoids. So this is where we're actually getting sulfate regulation and what they call regulome, which is, I guess, the set of genes that are involved in regulation. And so this is in humans now. So this is in human brain specifically. And so the abstract reads, self-organizing neural organoids grown from pluripotent stem cells combined with single-cell genomic technologies provide opportunities to examine gene regulatory networks underlying human brain development. Here we acquire single-cell transcriptome and accessible chromatin data over a dense time course in human organoids, covering neuroepithelial formation, patterning, brain regionalization, neurogenesis. So these are all these things that we looked at in the mouse, kind of what we're looking at in the human organoid. So this is from human-derived cells, then we're building organoids, and we're trying to identify the formation of the nervous system structures. We also identify temporally dynamic and brain region-specific regulatory regions. So this is where you want to know if things are forming, what the regulatory regions are being, activated, what regulatory regions are being activated, and that can tell us something about maybe brain development more generally. So they actually developed a framework called PANDO, which incorporates multi-omic data and predictions of transcription factor binding sites to infer a global gene regulatory network, describing organoid development. We use pooled genetic perturbation with single-cell transcriptome readout to assess transcription factor requirements for sulfate and state regulation in organoids. So this is something where there are genes for organoids. It's where what genes are being expressed and how are they being regulated and how does that create a stable organoid. And it may not be that they're, created, that they respond because they're organoids or not. It's just that they respond to that environment that the cells find themselves in. So that's something to be aware of when you're reading this. We find that certain factors regulate the abundance of sulfates, whereas other factors affect neuronal cell states after differentiation. So this is interesting that there's a sort of something that regulates the sulfate or maybe the ability to differentiate. And then there are other factors that affect neuronal cell states after differentiation. So some control differentiation and what kind of factors go into that. And other factors control the cell states. It's sort of like a homeostasis, keeping it stable. So we know this from a lot of cellular reprogramming literature where people are obsessed with figuring out how to get the thing to differentiate and then stay stable after it differentiates. Because you don't want it differentiating into, say, like a cancer cell or some other type of cell. You want it to remain your target cell. So this is something that we can learn from here as well. We show that the transcription factor GLI3 is required for cortical fate establishment in humans, recapitulating previous research performed in mammalian model systems. We measure transcriptomic and chromatin accessibility in normal or GLI3 perturbed cells. So this is again where we're doing not a knockout, but we're perturbing that factor in these cells. So we have a wild type and we have this, not really a mutant, but we're perturbing that gene, expression of that gene, and identify two distinct GLI3 regulums that are central to telencephalic fate decisions. So this is the telencephalic region of the brain. So this is one of the lobes of the brain where, I think it's the part where you have the that later you'll have neocortex emerge from. So this is something that where you have a regulum, which is just a series of regulatory regions and other things that coordinate this. So these telencephalic fate decisions involve a number of things. One of them is regulating dorsoventral patterning with HES4-5 as direct GLI 3 targets. So we're regulating patterning. And then one controlling ganglionic eminence diversification later in development. So this is where you have a diversification of regions in the brain. So this is where it's controlled, or these ganglionic eminence. I don't know exactly what that means. But basically it's the control of patterning and the control of regional diversification. But this is, again, is where you get this happening sort of early on when things are taking shape, and then later in development where things are sort of specializing. Together, we provide a framework for how human model systems and single-cell technologies can be leveraged to reconstruct human developmental biology. So in this case, we're actually using sort of a model of development. Again, we're looking at these brain regions that are forming, and then eventually they're going to form an entire organism. But we're doing this in a model, so we have to keep that in mind as well. They point out here is that direct comparisons between organoids and primary counterparts in mouse and human have quantified a notable similarity between the neural progenitor and neural transcriptome profiles. So this is a neuronal transcriptome, of course, is where you have the brain that's in place, and there's a mature brain, and there's a lot of transcription going on with respect to different functions and maintaining the brain. Neural progenitors, of course, have this sort of similar pattern of transcriptional activity. So, we can take neural progenitors out of context and we can observe this. We can also observe this in mature brains. So this is the sort of the justification of this kind of model, that we get the same pattern of regulation, if we have, whether we have our model or whether we have this in vitro or in vivo. So in vitro versus in vivo. Brain organoids have been used to successfully model microencephaly, which is where you get a very small brain, periventricular heterotopia, autism, and other neurodevelopmental disorders that may have differential effects in the various human brain regions. So the emergence of brain regions, you expect, changes in the transcriptome, but in brain organoids, we've been able to model these different disorders, so that means that the changes that occur in the transcriptome with respect to these different disorders also can be recapitulated in the in vitro models. But we don't understand the gene regulatory networks that coordinate early human brain development in normal and perturbed conditions. So we don't really understand the networks. We just understand maybe the profile, which means you just look at a bunch of genes, you look at how they're upregulated or downregulated, and then that's like a visualization, like we saw with the TSNE examples. So this is a difference because the regulatory networks are more predictive and they tell us things about how genes work together, but we don't know that. We're trying to figure that out. Research in model systems has identified core signaling factors and gene regulatory programs that orchestrate brain region formation in vertebrates. So we saw a little bit about that in the last paper. Initially, extrinsic signals establish an anterior-posterior axis. So this again, like in C. elegans, where you have the head-to-tail axis, where you get this, things are laid out in that direction, that triggers additional localized gradients downstream to segment the neural tube into distinct brain regions. So you start with a neural tube, it elongates, and then you get these brain regions that show up along there. It's sort of a segmentation process. Combinatorial activities of morphogens, including SHH, WNTs, BMPs, FGFs, and Notch, neuroregulins and spondins, converge on transcription factors So the transcription factors are things that regulate gene expression. You get these other morphogens that work combinatorially to execute regionalization. So you combine the morphogens with the transcription factors and you get different regional differentiation. Much of what is known about these pathways in regulating brain morphogenesis has been examined in non-human model systems, and it remains unclear how human brain development has diverged from our mammalian ancestors. So these are things maybe we can study in a model such as this. So this is a huge figure here where they look at a multi-omic atlas of brain organoid development, and it reveals developmental hierarchies and critical stages of fate decisions. So you can see here that you have these, you have this maturity over different days, you have the embryoid bodies, and then you get this organoid at day 61. So it takes 61 days to grow these things from the initial seeding of the cells. You get these embryoid bodies, and then you get these other structures, and then the organoids. They're just showing some of the examples of what people do with these different. This is a UMAP visualization here of the different cells. And then this is abstracting this into a graph. I'm not really sure a lot of the details of this figure, but let's see. Per A, of course, is where you have a schematic of the experimental design and UMAP embedding of integrated multi-omic metacells. They just show the different cell types that emerge from this. B is examples of loci with differential accessibility during organoid development from pluripotency. So B is actually down here. This is showing where you get these different genes and how these loci can be accessed during organoid development. So sometimes these loci that are on these critical genes that are necessary for, so this is PAL5F1, which is important in mouse development, and FGF8, which is also important in mouse development. This is VIM and DCX, and it just shows along this, along these loci, what's sort of the accessibility. So the chromatin will open up and allow accessibility to these genes at different stages in the development of these organoids. So from the PSCs to the ectoderm to the epiblast to the NPCs, the neurons, you have different levels of accessibility. C is schematic of the branch inference strategy. So they're doing some inference of these clusters, and they're actually building, I guess, lineage trees out of these. Although it's a graph abstraction, so it's then they assign them, they build a graph abstraction, and then they assign them to branches. So you can see here where you have these branches. This is differentiation in the telencephalon to different types of cells here, either non-telencephalon, dorsal telencephalon, ventral telencephalon, and so forth. So these cells go from precursors and they branch out in different directions, forming different substructures, sub-cell types, and so forth. So this is a pretty involved figure. And then this is their platform Pando, and it leverages multimodal measurements to infer a multiphasic gene regulatory network underlying human brain organoid development. So this is where this graph abstraction comes in. They're building these gene regulatory networks. They're showing now, instead of the cell types, they're showing the genes. They're showing that there's some key genes at the top here, and then they have these other genes further down in this regulatory network. You have SOX4, SOX2, PAX6. You have PAL5F1, FOXO1, and so forth, and they all kind of, have their place in this network. And so this is a regulatory network where they're using the PageRank algorithm to find the pathways through this network and how it upregulates and downregulates with respect to different parts of the brain. So this is where they can take this network and break it up into different functional regions with respect to different cell types and things like that. And then they show this global GRN where you have these different genes connected. So this is where they're really trying to get at this idea of a gene regulatory network. And then this one is where you have transcription factor perturbations and mosaic organoids. They reveal critical regulators of neurodevelopmental fate decisions. So again, this is where you have this, you're breaking this down into the different genes, you're perturbing different transcription factors, and you're looking for critical regulators, things that keep the cells stable. And so a lot of this analysis is based on that. And then this final figure we're going to go over here, is a single-cell multi-ohm view of GOI3 loss of function, reveals distinct regulums and effectors of dorsal ventral telencephalon specification. So you can see here that you have these different aspects of the multi-ohm, and we can do a lot of things analyzing it through a GRN and some heat maps. So that's all I'm going to talk about for those papers. Hope you found that useful.
Okay. Anyway, just a little bit of fun.
Yeah. Well, that was actually quite interesting. Yeah. So we might follow up on this in the future. We'll think about it some more, and that's great.
Yeah, while the world is falling apart, we do some fun things.
All right. Well, yeah. Have a good week, everyone. Thanks for attending.
Okay, thanks a lot.
All right. Thank you.
Bye.
Thank you.
Bye-bye.
Bye.
Yeah,
Yeah.
it was good.
Okay.
Oh, thank you.
Thanks.
Bye. Good.
Bye.
See you, Alon. Bye.